The population of many species is being adversely affected all over the world due to ill treatment as well as human destruction and modification of the habitats in which they live. This may increase the frequency of hybridization among taxa. On a wide scale, hybridization can threaten the genetic integrity or survival of any endangered species. Canis latrans, the coyote, Canis lupus bailey, Mexican wolf, and the Canis lupus rufus, the red wolf, are three native species of Canis and historically called Texas, United States home. Human impact was responsible for both Canis lupus species extinction in the wild, however they did survive in captive breeding programs. Reproductive isolation between all three taxa is evident based on morphological data. The red wolf was impacted by intraggressive hybridization with coyotes as it went extinct in the wild. However, the impact of hybridization on the other species in Texas is not yet clear. Variations at maternally and paternally inherited genetic markers were surveyed in coyotes from Texas, and Mexican wolves and red wolves from the captive breeding programs, as well as a reference population of coyotes from outside the historic red wolf range. Studies showed that hybridization had occasionally taken place between all three species, but the impact on the coyote population was quite small. This is shown by the levels or variation in the mitochondrial control sequence and the Y chromosome microsatellites, as well as a phylogenetic analysis. This phylogenetic tree shows the neighbor joining phylogeny of mitochondrial DNA control region sequences from coyotes from Texas, represented as LA, Mexican wolves, represented as LU, and red wolves, represented as RU. Bootstrap support is indicated on branches when over 50%. Each single asterisk represents a possible hybrid origin, and each double asterisk indicates haplotypes of clear hybrid origin. Based on the studies of mitochondrial DNA and the Y chromosome studies, integration has taken place, but the Y chromosome data is more complex and uncertain. This table shows the integration in the Texan Canis as indicated by mitochondrial DNA and the Y chromosome data. The results demonstrate that the influence motivating intraggressive hybridization in sympatric Texan Canis are numerous and rather complex. Body size and sex do not solely determine hybridization, and density-dependent effects aren't a full explanation of the observed pattern either. Although the hybridization between red wolves and coyotes apparently did not have a major impact on the Texas coyote population, it had and continues to have a major impact on the red wolf population. Hybridization with Mexican wolves may have had an important impact on red wolves' populace historically. However, this is no longer a risk to the red wolf now that the species are completely allopatric.